This is a continuation of the Chapter 6 lecture. This is Part 6B. So remember, we are discussing this idea of starting with a DNA template that provides the information on how to build a protein. And to do this, we have to go through both transcription, which is going from DNA to RNA, and then translation, which is the actual building of the protein using the RNA instructions that came from the DNA. So transcription creates a molecule of RNA from a molecule of DNA. And in a eukaryotic cell, this process occurs in the nucleus. So during the transcription, the DNA double helix unwinds. One strand of the DNA is used as the template to generate a molecule of RNA. And then in a eukaryote especially, the RNA is processed to become messenger RNA. And that messenger RNA is going to exit the nucleus via a nuclear pore, and then it will be ready to provide the information for the ribosome on how to make the protein. So just a couple review questions here. So let's talk about what actually is a gene. And you know, this might sound like a simple question, but it actually isn't. There's no simple agreed upon definition that accurately describes all known genes, okay? And if you end up taking Bio 152, we will get into this in much more detail than we do in 131. But for now, what we're going to define a gene is, it is a discrete unit of hereditary information consisting of a specific nucleotide sequence in the DNA. So we are going to define a gene as a piece of DNA, all right, that is made up of nucleotides linked together that is going to provide hereditary information. So, you know, one thing about DNA is, so everybody has DNA, right? All living things have DNA, but it's also very personal in a sense that no one in the entire world has the exact same DNA sequence that you have unless you are an identical twin. So, you know, there are these DNA tests now, you know, the question is would you get a DNA test? Would you get a personal DNA test before you had a baby? Or should we run personal DNA tests on newborn babies? There's a lot of ethical considerations that need to be considered when it comes to DNA information. So let's talk a little bit. We just finished talking about transcription. Remember, transcription is going from the DNA molecule to the RNA molecule. Now we're going to talk about translation, okay? Translation is going from the RNA molecule that, remember, leaves the nucleus, goes into the cytoplasm, and it's going to meet up with the ribosome ultimately to build a protein. So let's talk a little bit about ribosomes. Ribosomes are made from proteins and a type of RNA called ribosomal RNA, or sometimes it's called rRNA. So for this process of translation, we're actually going to talk about 3 different types of RNA. rRNA, which is ribosomal RNA, that makes up the ribosome along with proteins. mRNA, which we already talked about, the mRNA is what is transcribed in the nucleus and then leaves the nucleus and then goes into the cytoplasm. And then there's a third type of RNA called tRNA. And tRNA stands for transfer RNA. It's the job of the tRNA molecule to actually carry the proper amino acid to the ribosome so that it can be linked together to form the polypeptide, which will ultimately become the protein. So within each ribosome, there are binding sites for both the mRNA and the tRNA. So a couple things about the tRNA that's important. So the job of the tRNA is to carry the proper amino acid to the ribosome, okay? So the tRNA has two very important parts. So one of the parts is at one end, and that's where the amino acid is attached. That's what you see. This little purple circle here represents an amino acid, and in this case, the amino acid is threonine. The other part of the tRNA that's important is this bottom part, okay? And you can see it better over here. So remember, the mRNA is made up of 3 nucleotides at a time that code for a particular amino acid. And remember, that information is carried by the RNA, but before that, it was in the DNA, right? So the mRNA, the messenger RNA, is the carrier of that information. So it's really important that the right amino acid gets in place when you make the polypeptide. Well, that is with the help of both the mRNA and the tRNA. So they get together at the site of the ribosome. Here's the ribosome, okay? And what you can see here on the bottom in red is the RNA that is the mRNA. And remember, the mRNA has these codons, which are 3, actually, let me pick something that's a little bit better with red. So 3 nucleotides make up the codon on the mRNA, and that is coding for a particular amino acid. But then it's the job of the tRNA. So the tRNA is found, the anticodon, which is complementary to the codon, is found on the tRNA. So this anticodon is on the tRNA, and the codon is on the mRNA, okay? But it's very important that the tRNA anticodon will complementary base pair with the mRNA codon, all right? Because that's what's bringing in the proper amino acid, because there are different tRNAs with different anticodons that will have different amino acids attached, okay? Now later we're going to talk about mutations. And a mutation is just a change in the DNA sequence. Well, that change in the DNA sequence can change the RNA, the mRNA sequence. And sometimes when it changes the mRNA sequence, it will actually change the amino acid that is put in at the site of the ribosome. Okay? And that's because if the codon has changed, then the anticodon will change. And then the wrong amino acid can be brought in. So the genetic code uses these triplet codons. So every amino acid has a 3 codon code, okay? And if we know the sequence of the mRNA, we can actually determine the amino acid sequence using this chart. Now, students always ask me, do I have to memorize this chart? And the answer is no. You just have to know how to use the chart, okay? All right. So with the chart, it is a codon chart that it uses the mRNA codons. Okay, so let's say you have a codon that is A U G. Okay, and I know this is really messy, but I just want to show you how to read this chart. So you start where it says start here. Okay, so our first nucleotide is A. So we are going to start here with the A. Then our second is U. So we go from A, which is in this quadrant right here, to U, which is right here. Now we're just dealing with this quadrant, okay? So the third is G, and that's right here. So you go A, U, G, and here is the amino acid. It's methionine, okay? Now, it turns out that AUG is actually a special codon because not only is it the amino acid methionine, but it's also called the start codon. It's usually the amino acid that starts a polypeptide. So the question is, can you translate this code, okay, into an amino acid code? And to do that, you would need the chart, okay? And so, you know, what you might want to do is get your book out and get the chart that is found there. And then what you're going to want to do is go codon by codon. So we just looked up AUG, and AUG is methionine. Okay, then you want to look up the ACU and the AAA and the GAG and the UCA and the UAA. Okay, and see if when you do that using the chart, if you can come up with this polypeptide chain. So the process of translation creates a molecule of protein via the genetic code. Translation is going from RNA to a polypeptide or protein. And translation is divided into 3 parts: initiation, elongation, and termination. Okay? And translation begins when the ribosome is made up of 2 subunits, a small and a large subunit. And translation begins when those 2 subunits of a ribosome assemble on an mRNA. And then what happens is the tRNA then brings in the proper amino acid to match the codon, because remember that the codon on the mRNA has to complementary base pair with the anticodon on the tRNA. And that's going to bring in the proper amino acid. Ultimately, you have translation that results in a polypeptide. So here you can see each little purple ball is an amino acid. They are linked together by peptide bonds. And then remember that polypeptide is going to fold up into a 3-dimensional shape. So what happens in translation is the mRNA is read, the tRNA is going to continue to bring amino acids into to the ribosome. The ribosome's job is to link those amino acids together via peptide bonds, and the ribosome will continue to do this until it reaches what's known as a stop codon on the mRNA. When the ribosome reaches a stop codon, that is indicative that the polypeptide is done, and when it reaches the stop codon, the ribosome machinery will disassemble, and the completed polypeptide is now available to be used, or it can be modified, for example, in the rough ER, the Golgi, and it can be modified into a functioning protein. Now, with Mastering Biology, you do have nice videos that show the process of both transcription and translation. So in the next section, we're thinking about our opening question is, are you your genes? What are ways your physical or behavioral traits might be influenced by your genes, your environment, or both? Remember, we talked about in the last chapter, chapter 5, we talked about how genes provide information for physical traits, but that the environment also can play a role. So the process of gene expression is when a gene is expressed, it produces a product. And what we will discuss is that product will be a protein. And the process of gene expression is regulated in several ways. So when you think about it, all our cells have all the genes in the nucleus, okay? All right, so all the genetic information is in every cell. It's kind of like having a cookbook, okay? So this cookbook is going to have all sorts of recipes, right? It's going to have meat recipes and salad recipes and dessert recipes and breakfast recipes, right? Right. So the cookbook contains all those different recipes. But remember, at different times, you're going to use different recipes, right? I mean, you're probably going to make pancakes for breakfast, okay? You're probably, although sometimes people like to have, a pancake dinner, but the idea here is that every cell in your body has all the instructions to make all your proteins, but different cells will express different genes. Even though all the genes are there, not all of them are expressed in every cell, and not all of them are expressed in every cell at a particular, all the time, okay? So this idea of gene expression or gene regulation is the process of turning genes on and off, and that's a very important job of the cell. You don't want lots of proteins being made that you don't need, for example, okay? And so different cell types will express different genes. So, for example, not all cells need lactase, which is the enzyme that digests milk, right? Those are going to be in cells that are involved in digestion, right? Right? So different cell types will make different proteins, will express different genes. Now, there is something interesting about the X chromosome. So remember in chapter 5, we talked about we have 22 autosome pairs, and then we have 1 pair of sex chromosomes. And remember, if you're female, you have 2 X chromosomes. And the interesting thing is, in female mammals, which we are mammals, one X chromosome in each body cell becomes highly compacted and is almost entirely inactive. So this is an example of sort of extreme gene regulation, where for every cell, if you're a female and you have two X's in every cell, really only one is used to provide the instructions for gene expression. Now, when we talk about the way genes are regulated, there are several places along the path from DNA to protein that can be regulated. Okay? One way is during the process of transcription. During the process of transcription, there are some proteins called transcription factors, and these proteins must bind to DNA to actually turn transcription on, okay? And it has to do with, there is an enzyme that needs to bind to actually make the RNA during transcription, and for that enzyme to actually bind, you need these other proteins binding, okay? And that helps to control what genes are transcribed. Another way that gene regulation occurs happens after transcription. So after transcription in a eukaryotic cell, the RNA can be altered in several ways. So remember, transcription occurs in the nucleus, and before the RNA leaves the nucleus, it is modified. So there are chemicals that are added to one end, and that's called the cap, and also there are nucleotides that are added to the other end. And the other thing that happens in eukaryotic cells is something called gene splicing. Okay, and what happens is a gene is made up of, in a eukaryotic cell, is made up of two types of DNA, introns and exons. Introns may be removed, and the exons, which are what are actually expressed to make the protein, can be linked together after the introns are removed. Okay, so this is called splicing, RNA splicing, and this is a way that gene regulation can occur, because you can actually splice these exons in different ways to actually make different proteins. So thinking about gene regulation, gene expression, okay, we just talked about ways that transcription and what occurs after transcription when the RNA is modified to make mRNA, those are ways that genes can be regulated. But translation offers even more opportunities for gene regulation. The cell can control whether translation proceeds or not. It can control how proteins are modified after translation. Sometimes proteins will be made in an inactive form, and there needs to be further modifications before they are active. And then also the cell controls how long proteins hang out in the cell. Some proteins are broken down fairly quickly, while others can hang out for a longer period of time. So all this is further types of processes that can control gene regulation. Now, 6.9 is a section, one of those sections that we are not covering. So I will not test you on it. There are no review questions on it. So we'll be moving on to 6.10.